when thinking about what I would present today, of course, my first thoughts came to, let's talk about brain fMRI. But then my second thought was, you know, the last couple of years, we've also looked beyond the brain, still in the central nervous system. And if you go a little bit down, there is the spinal cord. And actually, the spinal cord has been pretty much neglected, for sure, in terms of functional imaging. So if you look just quickly at the number of publications per year, for whatever it's worth, if you look at brain fMRI, we're now leveling out at about 3,000–4,000 papers per year. This is logarithmic scale, right? If you look at spinal cord fMRI, we're about 2 orders of magnitude below. So we're reaching about, at most, 100 papers per year. And a couple of years ago, actually together with Silvestro Micera, we set out a bit the challenge, okay, we should, you know, supervise a PG student together and try to, you know, get the most out of spinal cord fMRI. And this started off first with one PG student, Nawal Kinani, who then stayed as a postdoc in the lab, and then, you know, went on with a couple of other PG students and postdocs. And so this is a bit like the MIPLAB spinal cord team that evolved since then. So I'm going to present you most of their results in a quick overview today. So first of all, when you think about the challenges of spinal cord fMRI, there are a couple. I mean, obviously, first of all, it's much smaller than the brain. So the diameter of the spinal cord is about 1 centimeter. It's a long and tiny structure. And so you want to have a high resolution in plane, but not necessarily between the different slices you position. And then you're much more sensitive to field inhomogeneity and the different susceptibilities of the nearby tissues. And you're also much more sensitive to the physiological noise contributions, especially the motion they induce. So I don't want to go in all the details of how you can do decent spinal cord fMRI, but there are a couple of things I like to quickly mention. First of all, compared to brain fMRI, you need to be a little bit more careful for the preparation. So you need to position the subject very carefully and actually make sure the curvature of the spinal cord, if we're talking about cervical spinal cord, is not too high. So you can use some pads for that, and in particular set pads, which also decrease the susceptibility artifacts. Just as with the brain, it's a very good idea to record physiological signals, especially respiratory and cardiac, because they will turn out to be very useful in subsequent steps. Then in terms of acquisition, if you're only interested in the spine, you need to go with the reduced field of view, and that means you will need to leverage sequences that either work on outer volume suppression or selective excitation. Then the slices are best positioned in a way they're perpendicular to the cord. Again, a reason to minimize the curvature. And of course, the shimming might be, well, is typically a concern because you want to have, of course, at least inhomogeneity along the cord as possible. And then last but not least, just as with brain fMRI, you need to take out your favorite toolkit of pre-processing steps. I don't want to highlight them all, but one of the most, let's say, distinct one with respect to the brain is that you will typically do slice-wise motion correction and also slice-wise artifact removal due to physiological noise. And in addition, because of the particular structure of the spine, you will also do smoothing in an anisotropic way and not in the typical isotropic way as we do with the brain. Now, luckily, over the last couple of years, a couple of groups over the world have put together all their efforts. And so we have now a spinal cord toolbox. There are also pipelines based on FSL and iLearn that start to actually standardize all these steps. And so we moved from the initial art way of doing spinal cord fMRI to a more scientific way. So when we started off with Nawal a couple of years ago, it's actually already 7 years ago, we said, let's do something very simple, you know, some basic mechanism, task fMRI, and we go on from there. So how do you do that for spinal cord? Well, something simple is, let's do some motor movements, and in particular, let's try to find some evidence that there is a rostrocaudal spinal organization, which means from top to bottom. And you can look at some different hand movements. So here you see there are 3 of them that we selected. And what you would then typically do is, based on the choice of those movements, you can look at the myotome, and you can map back the muscle activations that you expect from these specific movements to actually where in the spinal cord you expect activation. In particular, actually on the ventral side, because that's where the motor neurons are located. And so based on this known anatomical knowledge, you can predict what are called spinal maps, which you see here at the bottom left side. And so those are hypothetical activations along the spinal cord for these different movements. And so in a way, you can compare them with what you measure with fMRI. So that's the theory, because once you go in the scanner, and this is out of 19 subjects, the kind of activation maps group-wise that you get are shown here. They're a little bit disappointing. I mean, if you look carefully, yes, there is a bit more left and right and a bit more ventral than dorsal, and the differences between them seem to be suggestive for the spinal maps that we idealized first of all. But actually, the statistical significance of these maps is quite low. The inter-subject variability is very high if you dive into it. And there's actually a limited correspondence with this idealized activity that we set. So, okay, still we could publish this, but then what do we do next? And Nawal actually saw in the lab the work we do for the brain, and this was a stepping stone to bring that down to the spine, if I may say like that. And so let me do a little sidestep, and let's look a little bit more carefully at about what we know about brain fMRI. So the good thing is that if you start off with a hypothetical signal that we call the activity-inducing signal, which is in a way the neuronal activity, but at the time scale of fMRI, so at the second time scale. So I'm not talking about neuronal firing at milliseconds. So you kind of slow this down to a second time scale. Now, given that hypothetical activity, we know a lot about how that translates into the bolt signal that we measure with fMRI. In particular, there are known biophysical models, which are coupled differential equations, which tell us how blood flow links to volume, links to deoxygenated hemoglobin, and explain in the end the so-called hemodynamic response function, which we saw already a couple of times today. And this is a very sluggish function that takes 4 to 6 seconds to peak, and then about 10 seconds more before it goes back to baseline. So given that well-known characterization, you can now take again the activity-inducing signal. If you linearize this, make a convolution, and you actually have an idealized bolt response given that hypothetical activity-inducing signal again. And just in terms of signal processing, you can represent the linearized form of the HRF as a differential operator that has 1, 0, and 4 poles. Okay, that's nice. So because in reality, of course, we don't measure this ideal signal, but it's corrupted by noise at least, and probably also some model mismatch. But then maybe because we have the characterization as a linear differential operator, we can invert it. And maybe we can try to apply this inverse operator to the measured signal to get us back to this activity-inducing signal, or hopefully close to it. If you try to do that, that doesn't work. It doesn't work because this is a notorious ill-posed problem. And in essence, the forward operator is like a huge low pass. So the inverse operator is a high pass and just blows up all the noise, and you don't get back in a safe way. So the solution to that is to regularize the problem, which corresponds to introducing more assumptions. And the assumption we came up with is the so-called total activation framework. So here you actually see a real-time course from a resting state fMRI, posterior cingulate cortex. And if we apply the scheme that we developed, we are able to deconvolve that signal into an activity-inducing signal and even take the derivative of it into what we call an innovation signal or transient signal, which peaks indicate when you have a sudden change of baseline, if you want. Now, mathematically, that can be expressed as a generalization of the well-known total variation regularizer, which is essentially saying that the derivative of the deconvolved signal should be sparse, which you can express mathematically as an L1 norm of that signal. And there are well-known solvers to effectively come up with the solution to that problem, which is what we've done in the background now, and which leads to these solutions of activity-inducing and innovation signals. And so to bring it back to the neuroscientific level, we're actually saying that at the time scale of fMRI and the spatial scale of fMRI once more, we actually express the signal in terms of its transients. Okay, and those transients should be relatively rare in time. Okay, how rare, that depends on your regularization parameter, but you can see that there are indeed quite convincing looking fits to those time courses. Okay, so we leverage that a lot for brain fMRI, and let me now move back to fMRI because that's what, to spinal cord, sorry, because that's what Nawal did. And so does it work for spinal cord fMRI, given these rather disappointing results on task fMRI at the spine level? So here you see actually a time course from a gray matter voxel in the spine. It's clear it's more noisy than what you typically would observe in the brain, for those who are used to look at those time courses. But actually, if we do the total activation deconvolution, we actually get to something that looks interesting. And we can, of course, do the derivative because that's what the regularizer was driven by. And you know, we seem to express the signal in something that has transients that happen from time to time. And then again, just like we did at the brain, actually, we can do this for all voxels in the spinal cord. And then we can do temporal clustering that brings together the time points that show the same spatial pattern of transient activity. So if you look at that set of innovation-driven coactivation patterns, or ICAPs as we call them, I will show them in more detail, but they are, of course, localized patterns in the spinal cord. And because they're in the spinal cord, we like to call them spicy caps, spinal cord ICAPs. You see we're getting closer to the apero already. And so once you have them, you can then also look at their, you can actually bring them back, regress them back to the activity-inducing signal, out of which you can, oops, sorry, that was a bit too fast, out of which you can then extract some temporal features, such as the co-activation between them and the co-deactivation, which is essentially how they temporally overlap over time. Okay, so let me show you how that turns out if you apply this to the spinal cord. And there are, in a way, two regimes where this is very useful. The first one is in so-called low granularity. So you go for a low number of spicy caps. And the first thing we observed is that the spicy caps seem to be driven by a lot of activity. So the spinal cord in resting state is actually very restless, just like the brain, which was actually a first surprise, to be honest. Now, if you look at that, at those spicy caps more carefully, you see that they nicely show a rostrocaudal extent, so the functional organization from top to down in the spinal cord. And this is even so strong that we went to a data set that, as you see, has a much larger coverage here. And that you can see that if we keep this low granularity, we actually functionally segment the spinal cord. Now, maybe that might look as an almost obvious feature of functional organization in the spinal cord, but thinking about this a little bit more carefully, it's actually not that obvious because you have a well-known mismatch between the anatomy of the vertebral levels and the segmental levels. And so in an individual, it's actually very useful if you can, in a reliable way, come up with the segmentation of the segmental levels, because you cannot fully rely on the vertebral levels. And the more you go down in the spinal cord, the worse this gets. So you can use this technique, as you see at the bottom image here, for an individual where you actually segment the levels for a single person. And so this is an opportunity for clinical use because you can use it to get a personalized spinal fMRI segmentation, which you can then use for pain therapy or spinal cord injury, as we will see in a second. And it turns out that this is actually much more reliable than if you would try this with ICA. Okay, let's move from now low to high granularity. So we go for a much higher number of spicy caps. And the spicy caps you get are shown here. And actually, we have regrouped them in this different background color, orange and cyan, in descending and ascending components of the spinal cord. And you see that, again, for every level, we get a whole repertoire that seems to mostly repeat between the different levels. And they are very fine and precise spatial patterns. Now, notice that actually for this specific result, the data we used is the same quality as the one I showed you for the task spinal fMRI in the beginning, which looked very blurry, right? So this is one of the paradoxes I like to highlight here, because resting state spinal cord fMRI seems to contain a lot of information to come up with these very precise spinal segmentation, fully based on activity, while in task spinal cord fMRI, everything is mixed up. And so it means that the transients that drive this segmentation into spicy caps are sufficiently rich and different to come up with this very fine segregation. And you can actually, if you look carefully, there is a blue background on every spicy cap. That blue background is the best matching region from an anatomical atlas. So it's almost a one-to-one match with what we know from anatomy. Okay, and then because we can, thanks to spicy caps, look at the temporal activity more carefully, we could observe a couple of phenomena that were hypothesized for human, but mostly known from animal literature. So first of all, there is this intrasegmental coactivation, which is a well-known mechanism, and which we clearly see also if you look at coactivation from our spicy caps. And secondly, if you look between the segments, there seems to be more an inhibitory tendency, which again we can find as a deactivation between the spicy caps of different levels. So that was cervical level. In the meantime, we also tried this on data more low, so to lumbosacral spinal cord. And again, same story. You can use the low granularity to essentially come with a segmental segmentation, and you can do the high granularity to, even if the spine is getting very small there, to still get distinctions that are meaningful in terms of dorsal, ventral, left, right, and so on. And this actually brought us to a collaboration with Sergio from a NeuroRestore in Lausanne, where they were trying to use this for personalized planning of spinal cord injury patients. Now, of course, spinal cord is great, but we miss the brain. And that's why, thanks to a collaboration with McGill, the lab of Julien Doyon, we were able to get a very promising corticospinal sequence, which is proposing a big field of view where you can combine both the coverage of the brain with the cervical spinal cord, and actually everything in between. So also brainstem and cerebellum, of course. And everything boils down to a compromise. I think this compromise is actually quite reasonable. We have in-plane resolution of 1.6 millimeter and 4 millimeter interslice spacing, and still with the TR of 1.5 seconds. Okay, to say a quick note on the pre-processing pipeline, given how you need to deal with both the brain and the spine, things get a little bit more complicated because at an early point, you actually need to split your field of view into 2 parts and have a dedicated pipeline to properly deal with each of them. Okay, if you want to have just a quick look how this data looks like, so at the left-hand side, you have the T1 anatomical image, very nice. And at the right-hand side, you have a sagittal scan of a functional image, which I think looks really nice as well. And you see that the shimming does even if it's a large field of view and it's a difficult shimming case, does a very reasonable job. In terms of temporal SNR, obviously the spine has a lower TSNR than the brain, but still we get levels that are very workable for the types of analysis we want to do. Now, what can we do if we have both the brain and the spine? Many cool things. So first of all, we can look if we can infer spinal cord organization from the way it communicates with the brain. And what you can do is split the brain and the spine, doesn't hurt, and you can essentially now look at every voxel in the spinal cord and compute, in this case with the sensory motor cortex in the brain, a pattern of functional connectivity. So we're kind of getting for every voxel in the spine a feature vector, which tells us what is its functional connectivity with the sensory motor cortex. And then you can do clustering of the spine based on those sensory motor functional connectivity vectors. And if you look at the clustered connectivity matrix, you see that it, again, very nicely brings out the rostrocaudal organization of the spinal cord, but this time based on the way the spine talks to the brain. Okay, so it's not the spine with itself, it's how the spine talks to the brain. And this is very interesting because we all know the homunculus organization, which goes back to the 1930s with Penfield. But actually, this has been recently questioned in the sense that the homunculus organization in the sensory motor system might be actually much more, or a bit more complicated than we imagined from these early works in neurosurgery. And in particular, so what you see here at the top is from our data, what are like the top matching voxels in the sensory motor cortex, if you look at them per spinal level. And then we compared this with an external data set with task brain fMRI, and we looked at the best matches. And so if you look, for instance, here we have the face area in the jaw in particular, and then if you move down, you have the upper arm, and if you move further down, it goes to the forearm and the hand. And what you can see in these patterns that is different from the original Penfield homunculus is that there is like a mirror symmetric pattern, right? And that is what Gordon et al. In a famous Nature paper 2 years ago highlighted, is that you don't just have a linear organization in the homunculus, but something that is mirror symmetric and which seems to come out of our data quite well. Another application you can do, which is inspired from what people do at the brain level, is a spine print. Now, you already mentioned the fingerprinting. This is essentially spine fingerprinting. So is it possible to identify an individual from their resting state spinal cord fMRI? And so the question is, well, okay, and we use here the simultaneous brain and spine data. So if we only use the brain functional connectivity, this can be done very well. It gives more than 90% accuracy. If we only use the spine, we lose quite a bit. We go to 50%, but which is still largely above chance level. So it is possible to identify somebody from spine-only connectivity. And then you can do interesting games because you could say, okay, what happens now if we regress out from the brain the spine activity? And you see that there too, the accuracy drops from 90% to about 50%. So there is, in other words, a lot of activity in the spine that is meaningful to identify somebody from their brain activity. And vice versa, if you regress out the brain activity from the spine, you also get a huge drop, which is probably of no surprise. But still, it shows that there is a large degree of interaction and communication. And then the final result is, again, taken from the brain, where for those among you who maybe are regular attendees to OHBM, gradients, brain gradients is like a hot topic at the moment. So they are essentially low-dimensional embeddings of functional connectomes. And so if you try to apply this type of methodology to spinal cord fMRI, what you can reveal at the intra-segmental level is that the basic organization we know about left, right, dorsal, ventral is reflected in the intrasegmental functional connectivity patterns. Okay, so one final comment I like to make is somehow we made a tour. We went from the brain to the spine and back, bringing them together. And then also the way we did this was based on this total activation framework. Actually, if you bring the total activation framework also back to the original challenge we had with task spinal cord fMRI, what you can see is that it's actually very helpful as well. So what you see here is at the top, a time course, which is the original bolt measurement of a particular voxel that actually gives one of the highest significance. And at the right-hand side, you see the activation map at a certain significance level. What you see below is the same analysis, but now applied to the time courses that have undergone the total activation deconvolution. And so you see that the time course seems very much properly denoised. You start to recognize some of the activation blocks, and you see that the activation map thresholded at the same significance level shows a lot more significant voxels. Okay, so my take-home message for you would be that the spinal cord is more restless than you would imagine, and maybe that's not so surprising because it's our bridge between the brain and the periphery, and I'm increasingly thinking that it's supporting what I would call the bodily experience, which is all the time present and which should warrant a very intricate communication between the spine and the brain. So I think this has promises both for fundamental and clinical research. So let me thank the team again, in particular the spinal cord team I already highlighted, and the collaboration with the McGill people. And maybe as I'm the last speaker, let me also thank the CIBM for organizing this day. I think it was fantastic to see so many of you together. We are a very vibrant community, and we are very multimodal, multi-organ, multi-scanner, and translational from fundamental to applied clinical research. So it's absolutely fantastic, and I'd like to thank the whole organizing committee, Pina, of course, but also Sandra, Axel, Emily, Hamza, Elda, Sebastien, Olivia, Miguel, and probably many others I forgot. And of course, all the speakers and the poster presenters, and you, of course, for being here today and celebrating this all together. So thank you very much.